Hey guys, welcome back, and today I will be covering Unit 3, Biological Molecules. Like we do before, first let's go with the syllabus guide. If you all want, you all can pause the video and take a quick screenshot, and at the end of the video, I can check if all points have been covered. Right, coming down to the lesson, we'll be talking about four main things: carbohydrates, lipids, proteins, DNA, and practical tests done to identify some of these components. Before actually moving on to the components, we have some additional information. Carbon is an element present in almost all the biological molecules, and these carbon atoms join together forming chains or ring structures, which are known as macromolecules or polymers. So the terms monomers and polymers, where monomers are often the smaller repeating subunits that join to form a polymer. Now, as you can see in this symbolic representation, if we consider this whole thing as a polymer, each box would be representing a monomer that links together to form this lengthier polymer. Even as the definition suggests, monomers are constructed of repeating subunits, and polymers are long chains or macromolecules made up of multiple monomers held together by chemical bonds. Coming down to carbohydrates, which includes insoluble sugars like glucose or complex material insoluble ones such as starch. The simplest kind of carbohydrate is simple sugars or monosaccharides. So the composition of carbohydrates is made up of carbon, hydrogen, and oxygen, where the short form is CHO in the ratio of 1 carbon is to 2 hydrogen is to 1 oxygen. Generally, with the term carbohydrates, the word saccharides is also used, where saccharides give the meaning of sugars. As you all know, all sugars are mainly produced of photosynthesis, and most of them are often soluble in water, and usually most of them end with the suffix OSE. So those are the few main things that you have to remember about carbohydrates, especially the composition: carbon, hydrogen, oxygen in the ratio of 1:2:1. Moving on to some additional information about the classification of carbohydrates, where we consider the three: monosaccharides, disaccharides, and polysaccharides. Oligosaccharides isn't actually necessary for our level. So monosaccharides are sugars with a single carbon ring, as the name suggests, where mono means one. Then disaccharides has two carbon rings, where the prefix di suggests two. And finally, the polysaccharides, which are insoluble in water, are formed when multiple carbon rings or when multiple monosaccharides join together. So you guys might have to remember some of these examples under the three categories: monosaccharides, disaccharides, and polysaccharides. Under monosaccharides, we have glucose, fructose, galactose, and under disaccharides, we have sucrose, lactose, and maltose. And under polysaccharides, we have starch, glycogen, and cellulose. As you can see in the diagrams here, sort of a symbolic representation, where monosaccharides, a single carbon ring represented by a single plate, whereas disaccharides have two carbon rings linked together, which are symbolized with this diagram with two plates linked. And polysaccharides, multiple carbon rings linked together, as shown in figure 4.5. So that's it about carbohydrates. Next, coming on to lipids. First, you all should know how to differentiate between fats and oils, where fats are semi-solid or solid, whereas oils are liquids at room temperature. That's the main difference. And when considering the composition of lipids, they are made up of carbon, hydrogen, and oxygen, similar to the carbohydrates. And a fat molecule generally consists of a glycerol backbone and 3 fatty acid tails. And here again, I have given some extra information as well, where excess fat consumption leads to formation of plaque in arteries. These plaques are formed with a combination of lipids and proteins. LDL, which stands for low density lipoprotein. Although generally the liver produces LDL, when there is a high intake of fats, helps to raise this LDL level in blood more than the natural amount. And this increases the risk of plaque formation. Also have in mind that most animal fats form saturated fatty acids, whereas plant oils form unsaturated fatty acids. And these plant oils are considered to be healthier than animal fats. So when considering the structure of lipids, as I said before, lipids are made up of 3 fatty acid tails and 1 glycerol backbone, as shown in figure 4.8. So that is what you must remember when drawing a lipid molecule, whereas the actual composition of these fatty acid molecules and glycerol backbone is shown on the left, which is just some extra information so that you all have an idea about the real composition or what it looks like. So thirdly, coming on to proteins, where proteins constitute of carbon, hydrogen, oxygen, nitrogen, and sometimes sulfur and phosphorus. So the short form is CHON S-P. Have in mind that there are 20 different types of amino acids, which are generally soluble in water, and these amino acids are arranged in a particular sequence to form different types of proteins for a specific purpose. The shape of these proteins are very important because of the active site and for the antibody-antigen binding. This part you will understand more clearly in the enzymes lesson. So the smallest unit of protein or the monomer is amino acids. A few amino acids link together forming a polypeptide, and a few polypeptides link together to form the giant structure known as proteins. In the diagram, you can see the amino acid structure where it constitutes of an amino group, NH3, a hydrogen, a carboxyl group, and an R group, where this R represents a variant where different types of carbon components join to form different amino acids. Just have a small idea about this overall amino acid structure, because it might sometimes come in biology, but in chemistry as well, you will learn about this in polymerization. Right, so that's it about proteins. Now moving on to the fourth main topic, DNA. Each and every living cell has a nucleus except for bacteria, as they have no true nucleus. And in this nucleus is where you find the chromosomes. And in these chromosomes is where you find the DNA. This DNA molecule, when magnified, we can see its structure is more like a spiral and is referred to as a double helix structure. When considering the composition of a DNA molecule, it is made up of 2 strands of nucleotides that are linked together by organic bases. So if you zoom in this part, a nucleotide is a 5-carbon sugar molecule, a phosphate group, and an organic base. The sugar molecule, the 5-carbon sugar molecule that is found in a nucleotide is known as deoxyribose, and there are 4 organic bases, A, T, C, and G, which stands for adenine, thymine, cytosine, or guanine. Remember that the 2 chains of nucleotides are held together by the bondings between these organic bases, as shown here as well. And remember that A would always bond with T and C would always bond with G. These bases bonded in the DNA naturally form a sequence that provides a code which is used to determine the kinds of proteins made in cells, determine how cells, tissues, and organs develop, and is the cause for our own personal characteristics. That's it about DNA. Hope you all understood the structure of a nucleotide, which is a 5-carbon sugar known as deoxyribose linked to a phosphate and linked to an organic base. The organic base could be adenine, thymine, cytosine, or guanine, which are represented by the 4 starting letters. This structure is known as double helix, and this is held together by the bondings among these bases, where A would always bond with T and C would always bond with G. Right, next is the practical part of this lesson. First, let's talk about the tests done for carbohydrates. There are 2 main tests depending on the type of carbohydrate, where iodine test is done for starch and Benedict's test is done for all reducing sugars that include maltose, glucose, fructose, and galactose. As shown in the table, when for starch, the chemical reagent added is iodine solution, and when a few drops is added on the substance. If starch is present, the specimen turns blue-black color, and if not, the specimen remains brownish-orange, which is the color of the iodine solution. Similarly, for reducing sugars, which include maltose, glucose, fructose, and galactose, the chemical reagent is known as Benedict's solution, which is named after a person. We add a few drops of Benedict's solution on the food specimen and heat it over a water bath, generally at 90 degrees Celsius. And depending on the amount of sugar present, the color will change from blue to green, green to yellow, and yellow to orange, and finally to red. And if there are no traces of reducing sugars present, the solution will remain blue. As you can see in this diagram given here as well, blue means no reducing sugars present, whereas red on the other side means the highest amount of reducing sugars are there. Then for proteins, we undergo with the Biuret test, where we add the Biuret solution to the food specimen and shake it. If proteins are present, the color changes to purple, and if not, the solution remains blue. The Biuret solution consists of sodium hydroxide and copper sulfate, which can be harmful to human skin. And remember that Biuret is the name of a chemical and is not named after a person, unlike Benedict's solution. Finally, the test for lipids, also called as ethanol emulsion test. Here we add about 2 cubic centimeters of ethanol to a sample piece of oil or food and shake the mixture. Then we add a few cubic centimeters of water and mix it well. If fat is present, a cloudy white emulsion or layer is seen at the top, and if not, the solution remains clear. The logic behind this test is that fat is not soluble in water directly, so ethanol is added to dissolve it. And then water is added that breaks the fat ethanol emulsion mixture, forming millions of tiny droplets that would float and form a layer on top. Right, I hope you all remember the 3 tests. For carbohydrate starch, we have iodine test. If starch is present, turns blue-black. If not, remains orangish-brown. Then for reducing sugars, we have Benedict's test, where if there are no reducing sugars, remains blue. If not, it will turn from blue to yellow to green to orange to red. Then we have proteins test, which is the Biuret test, where the solution would remain blue if there are no proteins present. Else, if there are proteins present, it will turn purple. And finally, the fat test or ethanol emulsion test, where a cloudy white emulsion is seen if there is fat present, and if not, the solution would remain clear. And that's it for this lesson. Hope you all enjoyed this video. If you all think that this note used in this video was useful and would like to have a PDF copy of it, please visit the links mentioned in the description below. And please do not forget to like, comment, and subscribe to my YouTube channel. It would be a great help. Thank you and have a pleasant day.